This video is part of the lecture Plant Breeding in a Modern World in the scope of the course Applied Genetics at the University of Ostrava. Heritability is a very powerful tool for breeders and can dramatically improve the outcome of a breeding program if it is properly used. At the same time, if heritability is poorly understood or improperly used, it might be more of a foe to the breeder. So let's see some of the common mistakes and assumptions that are made for heritability. A very common mistake is to assume that if a trait has a heritability of 0, it is not genetically determined. This is blatantly wrong. A null or low heritability simply means that there is no genetic variation in the population, but the trait is still hereditary, so the phenotype is determined by genes. For example, after several generations of very intense selection, all the alleles that have small additive contribution to plant growth are eliminated from this population. The population is phenotypically uniform and also genetically uniform for size. However, the genes responsible for plant growth have not been eliminated. The population has simply lost all alleles but one that is now present in all individuals and transmitted to all of their offspring. Otherwise put, the gene for size still exists, but they're all monomorphic, meaning they only have a single allele in the population, and hence all the plants have the same genotype and the same phenotype. Next, the estimate of heritability for one population is often assumed to be the same for all populations of the same species. We have already seen that this is not true. Heritability is a parameter that is specific to the measured population in the specific environment of measurement. This can be intuitively illustrated in our population under artificial selection for plant size. We have one species that has been measured in the same environment, and after every generation of selection, the trait heritability decreases. This is simply because after each generation of selection, the undesired genotypes are eliminated, which decreases the overall genetic variance in the population relative to the. Inversely, the environment can affect the overall environmental and phenotypic variation in the population. So even if the genetic variation does not change, its relative contribution to the total phenotypic variation will be smaller, and hence heritability will be smaller. It is often assumed that the effects of the environment and the genotype are independent from one another. This might be true in some cases, and when it is true, it largely facilitates the estimates of heritability. However, there is another component that contributes to the phenotypic variance, and that is the interaction between genotype and environment. If we look at the effects of the environment in this example, we see that genotype 1, which grows very big in a moderate environment, has a significant decrease in size in the warm and dry environment. Genotype 4, on the other hand, seems to be barely affected by variation in the environment. If we do not account for this effect of the interaction between genes and environment, our estimates of heritability will be biased, because instead of containing the relative contribution of the genetic variance only, it will contain the sum of genetic variance and the variance due to the interaction between genes and environment. To avoid such problems, ideally the estimates of heritability should be made by growing the same genotypes in different environments so that the effect of interaction between genotype and environment can be estimated. This is often limited by available space and resources for the breeders. Estimates of heritability can also be biased if the supposed kinship between individuals is wrong. We saw that one way of estimating heritability is to use a parent-offspring regression, and that heritability is twice the regression coefficient. This is because on average, parents and their offspring share 50% of their genes, provided that there is no inbreeding. The 50% of shared genes comes from the fact that the offspring inherits half of the genetic material from one parent and half from the other. So the offspring share half of their genes with any of the two parents. The shared value of half is also called the coefficient of relationship and is marked as phi. We can also estimate heritability from a half-sib pedigree if we know the average coefficient of relatedness between the half-sibs. Let's see how that goes. So first, for our pedigree, we have two parents which will be unique to each offspring, that is parents 1 and 3, and parent 2 is common to both offspring. So we have half-sib individuals that have one parent in common and one different parent each. If both offspring inherit the same allele from their parents, their coefficient of relatedness will be 1/2 because they will share 50% of their genes. But offspring can inherit different genetic combinations from their parent in common. In this case, they have no alleles in common and their coefficient of relatedness is 0. Here, they have the white allele in common for a coefficient of relatedness of 1/2. And finally, they can have this combination where the coefficient of relatedness is again 0. So if we calculate this on average, the coefficient of relatedness of two half-sibs is 1/4. Therefore, heritability will be estimated as 4 times the regression between the half-sibs. But this is only true if none of the parents are related to one another, which was implicitly assumed in our calculation. Now let's imagine that parent 1 and 3 are also related. They both have this blue allele that was inherited from a common ancestor. In this case, their coefficient of relatedness will be 1/2. All the possible genotypes of both half-sibs are shown here, so you can calculate it on your own, or you can trust me when I say that in this case, the coefficient of relatedness of these half-sibs is 1/2. This coefficient is higher because the half-sibs have more possibilities to inherit the same genes, this time not only through their common parent, but also through the two parents that are different but yet genetically related to one another. So if we're not aware of the relatedness in the ancestry, we will wrongly assume that the coefficient of relatedness between half-sibs is 1/4 and then calculate heritability as 4 times the regression between half-sibs. Whereas if we know that the parents are related, then we can adjust the coefficient of relatedness of the half-sibs and calculate heritability as 2 times the regression coefficient. You can obviously see that multiplying the heritability with the wrong coefficient will lead to a gross overestimate. It is thus very important to be certain about the relatedness between individuals in a population before estimating heritability. This is fairly easy to do when breeding cattle, for instance, because we know exactly which cow and bull have been mated. But what about crops, especially the wind-pollinated ones? It is almost impossible to identify the mating partners in this case. Fortunately, with the development of new molecular techniques, it is possible to solve this problem using molecular markers to estimate the coefficient of relatedness between individuals. If the plants are genotyped, then we can calculate the genetic distances or relatedness between any two plants as function of the number of alleles they have in common on multiple molecular marker loci. For each pair of plants, we can calculate the degree of phenotypic similarity and the coefficient of relatedness. And if we correlate these two values in the whole population, we will obtain an estimate of heritability. So when using heritability to breed plants, there are a few experimental design caveats that should be avoided as much as possible. First, be certain about the pedigree or the coefficient of relatedness in the population. Keeping a meticulous pedigree of all past crosses is highly desirable when possible, and alternatively you can use molecular markers to estimate this pedigree if it's not available. Second, control for the environment. Make sure that the environmental variation is minimal to avoid inflating the estimates of heritability. If possible, it is also recommended to make multiple estimates of heritability of the same population but in different environments. In this way, the breeder can account for possible genotype-environment interactions, which can also inflate heritability. Once you are certain of the robustness of your experimental design, beware of misinterpretation of the results. Most important of all, remember that heritability is not heredity. Having null or low heritability simply says that there is no genetic variation in the population and any breeding effort in the population is useless. But it does not mean that the trait is not regulated by genes, so it can potentially be improved through other breeding techniques such as controlled crosses. Heritability is also a unique parameter for each population and environment of estimate, and if it is applied to other populations in different environments, it might reduce its predictive power or even render this parameter completely useless. Finally, because of all the possible caveats in the experimental design that cannot be fully accounted for, heritability, and in particular broad sense heritability, should be taken as the uppermost limit of the response to selection, because its estimate will probably contain other variance components that could not be fully eliminated, such as microenvironmental variation, genotype environment variation, dominance effects, and others. Despite all of these precautions, heritability can be a very powerful tool. If we precisely estimate it and properly apply it can predict the mean phenotype in a population based on the phenotype of the parents, and it can predict the response to selection way before the actual plants under selection have grown in the field.